Okay, let's move on to our next type of channel. These are ion channels. Ion channels are going to be pretty selective to only allow certain ions to transport across. The prototype that we're going to use is going to be the nicotinic acetylcholine receptor. But for ion channels to work, you need to make sure you have a concentration gradient. That concentration gradient can be established by another type of transporter, such as a pump, or you take advantage of that ion difference between inside the cell and outside the cell. There is usually a limited amount of time that an ion channel is open, and it usually requires some sort of activation type of process. But this activation type of process can be varied, depends on the ion channel. Some of them are voltage gated, which means there needs to be a small electrical potential difference across that cell membrane. A great example of this is in nerves, where you have sodium and potassium channels opening and closing. But they won't open and close until there's enough of an electrical difference across that membrane of the nerve. Other types of channels are ligand gated. These means that they need a specific signal to open. Sometimes it only involves one ligand, sometimes it involves two. But besides voltage and ligand-gated channels, there are a few others. Some of the different cells that you have respond to stretch. Some respond to mechanical deformation. So there are other ways to open up ion channels, but the two main ones are voltage-gated and ligand-gated. Our prototype, the nicotinic acetylcholine receptor, will use a ligand-gated channel mechanism. The other thing about ion channels that we always have to keep in mind is usually there's a selectivity filter in them. And this allows for only then one or a few selected types of ions to move through at any one time. So you will have sodium channels, you have potassium channels, you'll have calcium channels, you have chloride channels, and only every now and then we have a non-selective, for example, positive or cation channel occur. So let's go to our prototype now. This is the nicotinic acetylcholine receptor, and this is very important in transferring certain types of cell signals and certain ions. You need two ligands to bind, and these are two acetylcholines that will bind to the portion of the acetylcholine receptor. By both binding, it will open up that selectivity filter and therefore allow ions to travel through it. In fact, though, this is very selective to only allow positive ions to pass. Now, which positive ions are going to pass? In this case, we have three choices. Is it going to be calcium? Is it going to be sodium? Is it going to be potassium? For this, you have to think back into how these are partitioned. If you have a gradient across that cell membrane, you'll be able to answer that primarily sodium will go across or influx into the cell. Although there might be a little bit of calcium, the primary one will be sodium. It doesn't work to have potassium cross because the concentration gradient is going in the opposite direction. So where do you use this channel and why is it so important? Well, it is the primary channel to engage muscle contraction. So if your nerves want to tell your muscles to contract, you're going to have to use a nicotinic acetylcholine receptor. And this is how your muscles figure out, oh my goodness, we need to contract. It is also very important in ganglionic function. And what the ganglia are specific aspects just outside for the autonomic ganglia, sympathetic chain, just outside the spinal cord, or you might have parasympathetic ganglia that are located closer to an organ. And these are synapses in which one nerve stops, sends a chemical signal to another nerve so it propagates on. And these are in certain spots known as ganglia. So this nicotinic acetylcholine receptor are very important for both neuromuscular function and nervous system function.